This is a video of me. It's a new series, if this works out, explaining by IB biology topics within 30 minutes. Amazing hit. Start off the thing by, just quick flex. Okay, so this unit, topic 3, I break it down into, let's see how many parts. Let's hope I get this done in 30 minutes. I don't think I will. RNA, genetics, diseases, mutations, mitosis, meiosis, and genetic modification. I would say around 6 different sections. Let's start off from the beginning. I'm just kind of trying to explain it the best I can for people that missed it, the lectures or anything. So DNA and RNA. I think pretty much everybody knows this in my class. So this is gonna be a little bit of review, not too much. Codons, RNA, triplets, 3 letters, whatever, anticodons, triplets in tRNA, triplets, just 3 letters on DNA, mRNA, messenger RNA, transfer RNA, ribosome RNA. We didn't really cover ribosome RNA much. I'm guessing we're gonna go over that in the future, but whatever. Okay. The antisense strand, the thing copied during transcription, that's a little more obscure, but that's not really important. Okay, transcription. So this is transcription, translation, the main stuff, right? Most people do know how to do the transcription part where you turn an A into a U or whatever, but the steps, so RNA polymerase, that's the main enzyme in transcription. It unzips the DNA, kind of like helicased it, but it copies the antisense strand, and mRNA is made, and the T's are replaced with U's because there's no thymine allowed in RNA, only uracil. And then after transcription, basically the mRNA is processed. Introns, the removed extrons are made. The way I remember that is introns are interruptions. That's why you just get rid of them after. And then the poly A tail added at 3' protein cap at 5. You're not going to be able to know that. Not be able to. You're not going to need to, sorry. That's just protecting the gene, whatever, It's just like a shield almost. Okay, translation. mRNA goes into the ribosome. It's read by 3 letters at a time. That's like the golden rule for DNA. 3, codons, anticodons, whatever. And then the tRNA basically reads those 3 drops off that amino acid. You get polypeptides, right? Multiple amino acids tied together. One of the main things you have to remember is that start codon. It's OG. The way I remember it is like that gun, OG, for Call of Duty and stuff. This is so awkward. Okay, after translation, it's folded into alpha helix, beta pleated. That's whatever, You don't. There's certain stuff that I put on here you don't need to know, but it's just like the extra, Genetics, I personally hate, or not hate, but I suck at it. It's just a lot of vocabulary, really. The main stuff you need to know is Punnett squares, dihybrid crosses. Most people do know that, but I'll try to go over that, maybe a little bit, but it's just a lot of vocabulary, dihybrid cross, the way I think about it. It's a Punnett square, but complicated. So it's multiple alleles, right? And it's with gametes, I believe. And those are easy because it's going to be 9, 3, 3, 1. The 2 dominant traits, you're going to have 9 of. And then the 2, one's dominant, one's recessive, you're going to have 3 each, right? And then 1, both recessive traits. That's how I remember it. Dominant, 9, and it goes down from that order. Karyotype are pictures of chromosomes arranged by size or shape. Most people know that. Remember, we did the karyotype lab. You line them up by comparing which size, blah, blah, blah. Okay, and they show what... Sorry. They show if there's any genetic mutations, abnormalities, etc. within, at it's, what I mean. Anyways, With these karyotypes, you get them by you extracting the amniotic fluid, and then you arrange them. Not too hard to remember. That word is kind of weird. Amniotic fluid. Amniotic. It's a really cool word too. Okay. Alleles. This one got me so many times, so I forgot what it was. It's just different forms of a gene. So alleles are like the different variations, like uppercase A, lowercase a. Those are alleles, or different variations of that same gene. Gene, on the other hand, is a section of DNA on chromosomes. So DNA on chromosomes, it's located on chromosomes. But what it does is it controls the traits that are expressed. Alright, now genotype. This is a combination of alleles in an organism. It's just the alleles that make up a gene. That's all it is. That's the definition. This is why it's hard, because it's a lot of definitions. On this little study guide that I made, this is pure definitions. Phenotype is that expression. Gene, and you make that connection. When you see genotype, what's a genotype? Oh, okay, this is the combination of alleles in the gene, because gene. And phenotype is physical. Phenotype, physical. That's what's shown on the outside, the physical expression of the alleles. Homozygous alleles, they're the same. That's the definition. They're just the same. You have 2 uppercases or 2 lower cases. Did y'all see that? Am I being haunted? And yes, I do have Gordon Ramsay in the background, but I need to keep going so I can get this under 30 minutes. Heterozygous. There is a difference in the alleles. There's 1 uppercase, 1 lowercase, 1 lowercase, 1 uppercase. You know what I mean. It's gonna sound very weird, but when you think about the words, if you're struggling to remember which one's which, homo, same, and hetero, different, homosexual, like the same gender, hetero, different, heterosexual, whatever, right? That's an easy way you can remember it. Dominant, it's the trait that's expressed, it's the one that's shown, is strong, it's the dominant one. Recessive, they call it the mask traits. It's hiding, pretty much. It's a bitch. Co-dominant, both of the dominant alleles are expressed. My voice sounds so weird. I need some water. This is very awkward. Sorry. Co-dominant, I think most people would. You look at that word, there's 2 dominant alleles and they're both expressed, like black and white feathers in a chicken. If you guys remember that from Mr. Moore's class, or Macaulay went over it this year, but not much. Carrier, so they carry the allele. Remember we were talking about alleles? They carry one of the alleles, but they're not affected by it. They have it, but they're chilling, Have it, not affected. Carrier. Zygote. It's a single cell that's going to become a fetus. I don't think you have to remember that much. I don't think she's gonna. Hope she doesn't. But then again, the test is 10 pages. Okay. Gamete. Sex cells. That's all it is. Sex chromosomes, the 23rd chromosome, always 23rd. Whether it's X and Y, I think most people can get the difference. I don't think I have to go over that. Autosomal chromosomes, any other chromosome that's not a sex chromosome, Okay. You can go back to whatever you need to. Those are literally the first 2 topics, pretty much. We did DNA and RNA. Wasn't too bad, right? And then we've just finished genetics. That's a lot of vocab, but I'm sure you guys got this. I struggle with that. It's just a lot of memorization. But diseases. Okay. So there's 2 types of... There's a lot of diseases, but 2 types that we need to know. Sex-linked and autosomal. Sex-linked, obviously you can tell by sex. They're diseases caused by problems on the sex chromosomes. In sex-linked diseases, one of the main things you have to remember is only females can be carriers. So when you look at a pedigree chart and you see no males as carriers and you see some women as carriers, you're like, okay, this is a sex link because only females can be carriers. The 2 examples you remember, color blindness and hemophilia. Sorry, my throat is so dry today. Pause. Sorry, I normally don't even say pause, but whatever. It's just so awkward. I hate myself right now. Ugh. Anyways, let's keep this going. I wish I had like a whiteboard here. I'm gonna invest in a whiteboard so I can start writing down stuff and becoming an actual teacher and become dressed in like all suits and stuff. But okay, sex-linked diseases are always located on the X chromosomes. Y is a basic bitch. You don't have anything on it. Later on down the line, we'll learn about probably diseases that have to do with the Y chromosome. But right now, just think about it as it's only on the X chromosome. I don't know why I keep doing this. Okay. There's an autosomal disease. Their disease located on the autosomal chromosomes 1 to 22. And this is where both male and females can be carriers. If you see a male as a carrier in a pedigree chart, autosomal disease. So, the two main examples we need to remember is Down syndrome and sickle cell anemia. I'll be going over that later. I think literally the next one. Okay, yeah. Mutations. Okay. This is kind of the one that's interesting. Kind of. It's kind of heartbreaking too. But there's two types of mutations. Chromosomal and gene. Chromosome mutations. There's two. One you have to remember more than the other. The first one is deletion. Self-explanatory, removal of sections on chromosomes. It's really self-explanatory. Deletion of chromosomes. Anyways, non-disjunction. Cool word. But what it means is basically there's an extra chromosome or you lose one chromosome. It happens in anaphase 1 or 2. We'll talk about that a little more later. And an example is Down syndrome. You have an extra 21st chromosome. I'm going to go over why a little soon. It's going to all tie in. Watch, I got you. But non-disjunction, addition or deletion of chromatids. So you have an extra chromatid. Deletion is like removal of sections, parts of that chromosome. Gene mutations. This is the other type of mutation. There's also two types of mutations that we have to remember. Point mutation and frame mutation. One of them is a lot worse than the other. I'll go over that right now, obviously. Point mutation. This is the substitution of nucleotides. I swear something wrong with the light. You replace a U with an A or something, or a C with a G or something, right? You're substituting certain nucleotides. The thing with this is you could get lucky sometimes. A nucleotide could be substituted and you'll end up with the same amino acid. But there are certain examples where that doesn't happen, where it's unfortunate, like sickle cell anemia. In this, you see the amino acid glue replaced with the amino acid Val, resulting in a mutation, right? Frame mutation. Oh, I should have probably gone over more. I realized I didn't go over much of sickle cell anemia or Down syndrome, but just know sickle cell, 11 chromosome, amino acid glue replaced with Val. Down syndrome, you have an extra 21st chromosome. Not much to remember. Yeah, you're good. And non-disjunction occurs in it's fine. I'll probably go over it in meiosis maybe for Down syndrome, but sickle cell, just explained it. Mutation on the 11th chromosome. That's why it's not a autosomal chromosome. And amino acid glue replaced with amino acid Val. And it's a point mutation. Point mutation. That's one of the key ones. Point mutation. Now this is a scary one. Frame mutation. It's a weird name. But basically, it's adding or deleting a nucleotide. If you're having... Okay, the example that Ms. Mattel gave us, the cat sat. Sounds like a normal sentence. Now you're splitting it up into three letters, codon. So the cat sat, 3 letters each. Now imagine you just add an extra letter in there. And let's say G. That whole sentence is changed. It doesn't make sense anymore. So It's kind of like that. So this is one of those ones where if it happens, you can't go back. We don't need to know any examples. Sadly, I think it would be very interesting. But that's the main thing. It's really scary. A lot worse than point mutation, in my opinion. Well, obviously. Point mutation, you can get lucky. Frame mutation, you cannot. There's no way you get lucky. Mitosis. Mr. Mora. Okay. My voice sounds weird. It's because my throat's really dry and I'm losing my voice. It's not COVID. Interphase. This is where the cell basically spends its time to grow. It's like if you see the cell cycle clock, I think I have it drawn here. If I do, I'll show it right now. Right. It's a really terrible clock, and the mirrored thing you can't really see it well, but it's like that. It's like a good amount of portion of it. This is where the cell spends most of its time. It grows, whatever, and it's separated into 3 phases: G1, S, G2. These are pretty easy to remember, in my opinion. G1, G grows. It grows, right? S phase, DNA replicates. It starts getting ready for mitosis. And then G2 is when the cell gets ready to divide. Again, for mitosis. It's also where it's growing. The 2 Gs, that's where they grow. S is where DNA replicates, And then mitosis itself is broken up into 5 stages, including cytokinesis. Normally 4 stages, but we're including cytokinesis. The hardest one for me to remember, the hardest one for me to explain to people is prophase. Prophase has 3 main things you have to remember. I mentioned 4 because it just, I think you should mention all of them. But basically, at first, it's DNA. It's free-flowing, we learned about that. It's free-flowing. But then when you're getting ready for mitosis, then in prophase, that DNA that's all free-flowing, it starts packing up into chromosomes, right? So you have DNA condensing into chromosomes, and that nucleus that surrounds that DNA disappears because the nucleus doesn't like it when it's not DNA, like it's not just free-flowing. So it disappears, it goes away. It's like, see you later. Literally, see you later. And then the centrioles appear, and they start moving like opposite sides. They're like, you know what, I'm getting ready. And they start going to one side, one goes to the other, and they're ready, for war, pretty much. They kind of hate each other. Metaphase is the second stage. PMAT, by the way. That's the acronym PMAT. Prophase, metaphase, anaphase, telophase. And this is the M of PMAT, metaphase. And this one's really easy. Meta, middle. The chromosomes that got formed, remember, DNA condenses, they start lining up in the middle. And the centrioles that are formed, they release these things called spindle fibers. It's kind of fascinating. They release them and they attach to the center. They attach to the centromeres. It's a cool word. And it's a lot about the middle, It attaches to the middle of the chromosomes, the centromere. The chromosomes are in the middle, and all this. And that's the main thing you have to remember. Chromosomes lie in the middle, meta, and centrioles release the spindle fibers, which attach to the middle, the centromere. Pretty simple. Anaphase is the simplest one. Chromatids pulled apart. That spindle fiber basically just pulling on it and separates the chromosomes into chromatids, which is why chromosomes and chromatids are not interchangeable. Whatever. We'll leave my saltiness on the side. Telophase. This is where the cell begins to divide into 2. It starts going to have. You just pulled apart the chromatids, and those 2 separate things, they're different now. So now 2 new cells are beginning to be made. And that nuclear envelope that was like, see you later. It's like, hey, I'm back. And it starts to appear again around the chromatids. Yeah. And then cytokinesis, 2 new of the cells, they're divided, they're formed, yeah. Meiosis. Very similar to mitosis with its stages. The 4 stages: prophase, metaphase, anaphase, telophase, as I was saying earlier. And the difference is there's meiosis 1, meiosis 2. And it's really simple to remember the stages. Prophase 1, in meiosis 1, right? There's prophase 1, metaphase 1, anaphase 1, and telophase 1. And in meiosis 2, there's prophase 2, metaphase 2, anaphase 2, and telophase 2. Okay. And the main things you have to remember, because Macaulay didn't really go over like the individual steps, so I would assume they're kind of similar to mitosis. But the main things you have to remember: prophase 1, literally the most important thing, crossing over. Okay, so this is where homologous chromosomes. Sorry for everybody that I took the wrong answer to on the test. This is where the homologous chromosomes exchange genetic material by almost crossing over, and they share that, and that causes genetic variation. It's why we're all different. It's why I look different from the person watching this video. Whoever that is probably gonna be nobody, but whatever, what's it called? It's how I look so much different from, I don't know, Macaulay. There's other reasons. You know what I mean? It's so important because that genetic variation. Anaphase 1 and anaphase 2, these are the other things. Remember how I was talking about non-disjunction? This is where non-disjunction happens. Remember how in anaphase, the chromatids are pulled apart, right? The spindle fiber starts pulling, they're pulled apart. And non-disjunction is basically mutation where a mistake happens while it's pulling. There's an extra chromosome that gets pulled to one side, or the one side loses chromosome because there's now 3 chromatids on one side, or something like that, right? I hope that makes sense. If it doesn't, you can always text me. I don't know how well this video is going and how informative it is. I hope it is. I could try to make it simpler, but I think you also know how you also need to know how to write it on the test. Hope. I don't know. Mitosis versus meiosis. There's it's gonna probably ask you a question on the test. Outline the differences between meiosis and mitosis. What you have to do is you have to make a T-chart. You can write it down in sentences, but I think T-chart's easier. In mitosis, you're forming identical cells. In meiosis, you're forming non-identicals. Remember the genetic variation? That's why they're not identical. And in mitosis, you're forming somatic cells, known as body cells. But in meiosis, you're forming gametes, the sex cells. That's a main difference. Mitosis, you're forming all the cells you need to grow as a human, and then meiosis are your sex cells. And because of the sex cells, in meiosis, there's 23 chromosomes, and it's a haploid cell that's been produced. Because remember, I don't think I went over this earlier, but haploid, diploid, haploid's half, diploid's double. I didn't go over that. I apologize. I probably should have planned this out better, but I hope it's okay. I'll do better next time. Mitosis, you have 46 chromosomes. It's diploid, and one of the main things, 2 cells are performed. To go over that, 2 cells are made. They're identical cells. They're body cells, and they're diploid with 46 chromosomes. Meiosis, on the other hand, non-identical cells, 4 cells, haploid cells, and they're gametes, sex cells. Okay, damn, this is the last one, and then I'll go over some other things too, but whatever. Genetic modification, pretty interesting, but pretty scary. So these are the 3 tools, stuff you'll use in genetic modification. You have the PCR, it replicates DNA. So, DNA. Basically, in the PCR machine, what you're doing first is you're adding heat. And if you remember, heat denatures a protein. And in this case, heat's going to denature the DNA, which is not the correct term because you don't denature DNA. But let's just say it, And then that heat basically breaks the DNA into 2 different strands. Then you add the primers. You know how Ms. McCowie always talks about the painting, and you add a primer before. It's that. The primers, they attach to opposite sides. They prepare. Primer, prepare. And then Taq polymerase, the polymerase from the bacteria, they copy the strands. In 3 words, it's called denature, annealing, and elongation. Denature, you're separating the 2 strands. Annealing, keeping it from coming back. And Taq polymerase, you're elongating it, you're replicating it, right? That's PCR. Restriction enzyme, easy. It's an enzyme, cuts DNA at specific sites, wherever you want it to, pretty much. Gel electrophoresis. Hate this shit. Okay. So it's basically a machine which, after the PCR and the restriction enzymes, you're cutting all this is what separates the DNA pieces that are formed by the restriction enzyme. And it separates them according to size and charge. The smallest base pairs are going to travel the fastest. You know how they always say, like, the shorter you are, the faster you run, or something. Like, the smaller you are, the faster you are. It's like that. The smaller base pairs, you're going to go farther. You're lighter. Right? You have less weight to carry. DNA is placed on the negative side because DNA itself is negative, and it's going to travel to the positive. Opposites attract, nature, physics, I don't know. I'm not a physics nerd, I'm the bio nerd. Okay, so that's all it is, gel electrophoresis. Those 3 main tools you have to remember. PCR, restriction enzyme, and gel electrophoresis. Why am I doing this? Okay. The steps to form like insulin or H factor for hemophiliacs. Basically, if you want to replicate something, right? You're gonna get that desired gene. Getting that desired gene, or identify it, or identify the desired gene. You're cutting it with the restriction enzyme. The restriction enzyme goes chop chop, and it gets that desired gene. Now you're gonna put that on the side for a second. You're gonna use that same restriction enzyme, get a bacteria plasmid, and you're gonna cut that same. You're gonna cut it using the same restriction enzyme. Now you're going to place that gene into the plasmid. You put the plasmid back into the bacteria, and it's going to start multiplying, start going to make, and you're going to make that desired gene. It's really interesting. If I wanted to replicate, I don't know, my hair, because this hairline's receding. Oh, we don't need to see that. I could, you take that desired gene, you cut it, you get the bacteria plasmid, use that same restriction enzyme, cut it again, put that gene that I want into the bacteria, or into the plasmid, I apologize. Put that plasmid back into the bacteria, and you start letting it multiply. It starts forming. It's so cool. Okay. And then the last thing, cloning. Simple steps. This is the simplest I can make it probably, and instead of going, first you obtain the novocyte from a female, no. Basically, what you're gonna do is you're gonna get a female egg. You're gonna remove the nucleus. Just get it rid of it. It's a blank egg, right? Now, whoever's gonna be the clone, the person getting cloned, right? If it's, I don't know, shout out to Jacob. We're saying he's gonna get cloned. We take a cell from him, and we take that cell. We take that nucleus of the cell, place it into the female egg. You zap it with electricity, right? And people are debating on how long you zap it for, but whatever, that doesn't matter. You take that and then place it into surrogate mother. You're done. That's all it is. It's really that simple to clone, but illegal. Yeah, because centromeres, crazy things. Not centromeres, sorry, telomeres. And that's pretty much the whole topic. I hope I was able to make it simpler. If I wasn't, you can text me. And that is literally perfect. 29 minutes and 45 seconds. That is genetics and DNA and RNA and stuff. Hope this helped. And I'm waiting for it to hit 30. Alright, let's 1 and hit 30. Okay. There we go.